Then uh, then you gave us a brief about the food processing, like what are the different purposes of food processing, and you made us familiar with the trends in food processing industry. Then you gave us an approximate flow of how the food processing works, like ingredient preparation, scaling for formulation, blah blah blah. And then, sir, there you introduced the typical steps in the food product development. Uh, like it is an iterative process, so you talked about that. And then you told about some metrics through which you can judge the food quality. And you also mentioned how the food quality goes down through the various stages of the process of the supply chain. Like when it is plucked, it is of high nutrition value, but finally when it goes into our stomach, the value reduces so much. And you also finally you talked, I guess, about the undesirable changes in quality attributes like the texture, flavor, color, and nutritional value changes undesirably. So you talked about that as well.
Okay, very good, very nicely narrated. Mr. Sitaram, did you hear Mr. Sitaram? क्या बात है, ज्वाइन करके लोग नहीं है? सीताराम, you are muted. You can unmute and then speak. He will not speak. I think he is not there. He has joined and he is not there. Okay. All right. I think still many people are not joined, but anyway, let me download the attendance now. Give me 2 minutes. Okay. All right, it is saved now. Give me time to share my screen. I think you can all see my screen.
Yes, sir.
And see the PPT, and that you told that I think we were here undesirable changes in the nutritive value, etc. That we discussed. That we discussed this food quality loss reactions, and then what are the undesirable changes in quality attributes and undesirable changes in nutritive value. All right. Okay. So now let's proceed further. So these changes, that is whether undesirable or little later I will tell you some desirable changes also. Okay, which we do in getting the food processing. We process the food to encourage the desirable changes in it. We preserve the food to discourage the undesirable or spoilage related changes to it. But all these changes, they are mainly because of the one or other type of the reactions leading to food, reactions taking place in the food. Okay. And the major chemical and biochemical reactions, and many of them are influenced or supported by microbial processes. So the reactions which can cause deterioration of the quality of the food, particularly, they are listed here. Major reactions, that is the non-enzymatic browning, enzymatic browning, that is when you peel an apple or potato or peel a banana and leave in open atmosphere, then its surface becomes dark, its surface becomes brown. So that is the case of enzymatic browning. There are certain enzymes and substrates, and they instant in the presence of atmospheric oxygen, instant oxidation reaction goes, and they go for enzymatic browning. Non-enzymatic browning, you can see when you these good examples are cake, biscuits, etc. That is the brown color of the crust in the bread or brown color developed during baking. These are all because of the non-enzymatic browning reactions, then where that the sugars and amine, they, when the food material are heated, then the free sugars and free amines, they react with each other, and a series of complex reactions takes place, and ultimately they form a new carbonyl compounds, okay, which have saturated, unsaturated double bonds, which absorb light of different intensity, and they appear brown in color, brown to black. So these are similarly lipid hydrolysis or lipid oxidation or reduction, protein denaturation, protein cross-linking, protein hydrolysis, or oligo and polysaccharide hydrolysis, polysaccharide synthesis, degradation of a specific natural pigment, like the chlorophyll is the green pigment in the vegetables, and when these vegetables are heated or thermally processed, so there is change in the structure of the chlorophyll. Okay, sometimes the vital detaches is the vital group attached with the chlorophyll enzyme, detaches it with the vital group which is attached with the chlorophyll, or it removes the in the chlorophyll magnesium ion which is there. So because of this, there are various unstable or stable compounds are formed. That it means structural change in the chlorophyll, which gives the change in the color. That's why you see when the green vegetables are heated, the colors becomes either, I think, brown or dark or red. And similarly, there are many other reactions. This is whole the subject of food chemistry. I think some of these maybe Professor Goswami carbohydrate related, lipid related he will discuss, and protein related something I will discuss in the next classes, two other classes. Okay, so these are the major reactions, and each reaction class can involve different reactants or substrates, like for example oligosaccharide when we talk about, polysaccharide when we talk about polysaccharide, maybe starch, it may be pectin, it may be other glycogen, it may be other compounds, but the depending upon the specific food and the particular conditions for processing and storage. But they are called class because in the each class there might be several members of these several compounds, which can be called as polysaccharide, which can be called as protein, which can, in the protein there are various type of protein, in the lipids there are different types of fat, etc. So they are the reaction classes because the general nature of the substrates or reactants are similar for all the foods. So as I told you, non-enzymatic browning reactions of carbonyl compounds which arises from diverse reactions like oxidation of ascorbic acid, hydrolysis of starch, etc. And these all may result into the formation of carbonyl compounds, and this may result to the browning of the product. Lipid oxidation may involve primarily the triglyceride. That is, all the lipids are triglyceride, but depending upon the type of nature of the fatty acid attached in the glycerol with the glycerol molecule, there may be different types of differences in these triglycerides. And one food may contain one type of triglyceride, solid fat, other mostly the plant oils, they are mainly the liquid, etc. Animal fats are solid, and these are all triglyceride. But there is variation in their fatty acid composition, saturated, unsaturated, etc. So there may be all these differences, but the major process of change may be almost similar, whether it is more unsaturated fatty acid, less unsaturated fatty acid, etc. When we talk about rancidity, auto-oxidation of the unsaturated fatty acid is the primary event. So there may be large number of complex reactions which may take place, and these reactions briefly is summarized into this diagram. You can see, if we see the three major component of a food, there may be a lipid, full carbohydrate, and protein. Other components, of course, are minor amount, Of course, water is there, but the organic compound main are the three: lipid and carbohydrate and protein. So when the food is handled, processed, or during storage, all right, it may come across, food may come across to various factors. Like then accordingly, when it interacts with the different causative agents, different factors, then there are some changes in the original structure, original nature of the compound, like major compound, major bioactive biochemicals, etc., takes place, and a series of reactions following, as I told. So in the lipid, when it comes in contact with oxygen or heat in the presence of catalyst, etc., then these lipids are converted into peroxides. And these peroxides normally, once its reaction starts, then it is go for auto oxidation, and this reaction continue until unless some proper care is taken to prevent these reactions. Okay. Similarly, in the carbohydrate, when it comes in contact with heat, strong acid, or strong base, then these carbohydrates can be converted into reactive carbonyls, carbonyl compounds, which are having free reactive groups. Similarly, proteins, they can also be, depending upon the various conditions, they may convert to the reactive carbonyls. So now you see the peroxides, they may also interact with reactive carbonyls. For proteins, directly they may interact with the pigments and flavors and vitamins, etc. So these peroxides form, they may be react with even proteins, etc., interact with proteins, and then peroxides may be, proteins may be oxidized. So these various combination and permutations may take place, and ultimately there is changes in these pigments, vitamins, flavors, and original nature of the lipid, carbohydrate, proteins, etc., and which ultimately is manifested in the form of change in the color, flavor, nutritive value, or whatever you talk. That is the quality of the both nutritional quality, sensory quality, even physical form, textural characteristics, all these things are influenced. Okay. So let me, but even the same changes, let me tell you, don't get confused that these reactions which we talked about, various reactions, etc., many a times these reactions, that is, it are the same carbonyl compound reactions, organic reactions, etc., which also are responsible for the desirable change in the change in the quality attributes of the food. So only thing you have to find out, and in which food, under which condition, this particular reaction is desirable, this particular reaction is not desirable. Like for example, enzymatic browning, in many foods, it is desirable. The same reaction which causes the change in the color, in the case of tea processing, that is a desirable, highly desirable reaction. But in the case of fruits and vegetable processing, drying, etc., it is undesirable. Okay, so there are so many desirable changes through the same reaction route or some other new reaction route occur in the food during processing, and these changes can influence sensory properties, functional properties, or nutritional value. So, and there a few such changes, that is, which are encouraged during processing. Okay, that is maybe development or preservation of pleasing colors and flavors of food, improvement or preservation of texture of food, improvement of the functionality of food ingredients, inactivation or control of enzymes and microorganisms. When we heat the food, cook the food, so enzymes are inactivated, microorganisms are killed, so food has a better shelf life. Okay. Inactivation of anti-nutritional substances and other approaches to improving the nutritional value of the food. So these are some of the major changes. Of course, there are many other, but major class of changes which influence or which are considered to be desirable change in food quality during processing. Okay, then we will take up. I will give you briefly one or two examples in each case. Okay, so let us say the example of development or preservation of pleasing color and flavor in the foods. Okay, like you see the processing of food tissues like meat, coffee bean, nut, olives, coffee. I don't know whether you have how many of you have seen the coffee when it is brought, and that is the coffee seed from the when it is harvested. This coffee seed is green in color, but it is roasted, and roasting develops that dark color. Maybe in this subject is time permit. The latter part of the semester we will discuss coffee processing, etc. if possible. Then similarly, meat, you see the muscle when it is that is slaughtered. What is the type of this color, flavor and color of the meat? But after cooking, after processing, after this becomes. There are many such examples. Similarly, processing of fabricated foods like bakery products, confectionery products, snack foods, breakfast cereals. You see the wheat flour, even wheat flour, if you see that it is converted into different products, bread, biscuit, cakes, and all those such of bakery products. And all these products, they have different color, different flavor, all textural characteristics, etc. So all these changes, of course, are arise and are a result of the various chemical and microbiological processes, etc., and which people like it. In similarly, fermentation, cheese. In the case of alcoholic beverage, you see the how the grape juice is converted into a alcoholic beverage, and the color as well as flavor from the white grapes, even red color, red wine is prepared. So there are similarly in the post-harvest ripening of the fruits. You see green fruits becomes that yellow becomes other red color, and all those things, mango, etc. are good example of this. Similarly, disruption of the plant tissues. Okay, it results the good color change, etc. Then preservation of color and flavor is often achieved by either sometimes chemicals are added into the food, antioxidants, etc. Okay, or sometimes the conditions or processing conditions are managed in such a way that some undesirable components are removed. All right, or sometimes some permitted colors or permitted flavors, etc., are added. That can also be done. Or sometimes even the food is processed without any addition, without any deletion, but just maintaining the proper conditions, maintaining the proper factors, temperature, pressure, etc., etc., as so as to allow the desirable reaction in a proper manner. Removal of undesirable components. It is one very good example is the drying of egg white drying, that to convert into egg powder. And egg white contains glucose. So if this glucose is removed from the egg white by sometimes there is the microbial certain enzymes, etc., are added, and which utilizes the glucose, and therefore the this egg white, when it is dried, it you get a completely white color product. But if this glucose is not removed, then glucose will cause non-enzymatic browning. That is the protein of the egg and this glucose, they will interact with each other during drying process, and then it will cause non-enzymatic browning. The egg powder will become dark brown in color. So there are similarly many examples in this case. Similarly, improvement or preservation of texture. That is some of the example in this case include that softening of the plant tissues by heat. When you see the vegetables, plants, tissues, when you cook it, when you heat it, they become soft. So texture becomes texture which was earlier hard, it becomes soft. Rice after cooking, it becomes soft. Okay, forming of plant tissues through the action of calcium and endogenous pectin methylsterase. Okay, this pectin methylsterase is intentionally decompartmentalized and activated by the mild heat treatments, because These this is it prevents the hydrolysis of the pectin, because during this ripening, then pectin is hydrolyzed, so pectin is solubilized and tissue becomes soft, it softens. Okay, so there are various ways by which pectin is decompartmentalized, or it's maybe deactivated to prevent the tissue becoming soft. So it remains. Similarly, you see that sometimes even sugar is added, sugar syrup is added, etc. You get firm texture, tenderization of meat by addition of proteases. Sometimes the animal before slaughtering, it is injected with enzyme proteases like papain, etc. Or after slaughtering, the hard pieces of the meat, they are tied with the papaya leaf, or they are dipped into the papain solution. Papain is a proteolytic enzyme, so it breaks the larger protein molecules into a smaller one, and the hard pieces of the meat become soft. Okay. Similarly, there are meat relax, etc. By texturization, we get desirable texture. Gelling, coagulation, or firming of egg product. You see, when the raw egg, when it is boiled, okay, it becomes a solid, etc. That is also a very good example of the changes in the texture of the food material during processing. Formation of cheese by the development by fermentation or addition of acid to milk. When the milk converts to curd, you see the liquid milk, it becomes solid or semi-solid curd. So many. So hydrolytic reactions, many of which are enzyme catalyzed, appear predominantly among the processes that cause softening of the texture. Okay. If you say the example of the functionality of food ingredient, that is many times when we process the food, its functionality, its functional characteristics, that is the other, how it improves the processability or the eating quality or nutritional value or how it behaves inside our system or during the processing system. Many times it increases. Like, for example, heat denaturation of whey protein in dried milk intended for bread making, pre-rigor freezing of meat intended for sausage making to enhance its water holding capacity. Okay, it can hold more water. Aeration in the functionality of starch by gelatinization or chemical modification. Okay, when whipping is done, then air is incorporated and it gives viscoelastic properties and other properties are improved. Alkali processing of soya protein to impart new textural properties. Control of thiol disulfide interchange reaction in gluten to develop proper rheological properties in bread dough, are increasing the sweetness of corn syrup by isomerizing glucose to fructose. So this is sweetness, which is a functional characteristic here. So there are many examples. Another, that is by when you process many cases, that is the anti-nutritional in the food which you grow naturally. There are many anti-nutritional anti-nutrients present in the food. So in the processing, these anti-nutrients either they are removed or they are inactivated, etc. by one or the other method, and the quality nutritional value of the food is increased. A few examples of this include that is the naturally occurring anti-nutrient substances like trypsin inhibitor, hemagglutinin, etc. is in legumes, in soybean. One is the major, these are the trypsin inhibitor, hemagglutinin are the major factor. So, but similarly, thiaminase in fish, avidin in egg, phytates in cereal grains, etc. So these are some of the anti-nutrients that like avidin in egg. When you consume raw egg, this avidin goes and it binds the biotin, which is it is an essential vitamin. So it binds the biotin, and biotin becomes unavailable for its normal function. So it results into the malfunctioning of the body process. Similarly, trypsin inhibitor. Trypsin inhibitor is a biologically active substance. It binds the trypsin. Trypsin is a digestive enzyme. Okay, protein digestive enzyme which is present in our system. Okay, in the intestinal tract, gastric. Okay, so when you take raw soybean or such other cereals which contain these type of inhibitors, then it goes and it binds the trypsin. Means trypsin becomes unavailable for its process act normal process, that is the protein digestion. So protein goes passes undigested from the small intestine and it goes to the large intestine undigested, where microflora, microorganism find a good substrate, they grow on it, and there is a various problems like stomach ache or even that flatulence problems and many other things. Similarly, there are very phytates in the cereal grains, etc. Okay, they are considered to be anti-nutrients. So many anti-nutritional substances are proteinous and therefore are subjected to inactivation during moderate heating of moist food, like avidin, even thiaminase, trypsin inhibitor. When they are heated, soybeans are boiled, or when they are heated, these anti-nutritional factors are inactivated. In other instances, removal or chemical modification is a feasible approach, like enzymatic hydrolysis or removal of lactose from milk, that is intended for lactose intolerance people, because there are certain people which cannot digest lactose and that creates problem in such persons. So in that, there is from the enzymatic hydrolysis, the lactose is converted into lactic acid or some other compounds, and it is not, its form is changed, so people find it digestible. Similarly, reduction of stachyose and raffinose. Okay, stachyose and raffinose are higher oligosaccharides which cause flatulence problems, etc. Okay, so when we, and particularly these are the problems in the pulses, they are found in pulses. So if you uncooked pulse, if you eat, so you are bound to suffer from the flatulence problems and all those things. So these, during cooking, these stachyose and raffinose, because they have the 1,6 linkage. All right, and 1,6 linkage, our system, we don't have that enzymatic machinery, molecular machinery to break 1,6 linkage. So during cooking process, this 1,6 linkage is broken, and then you can similarly removal of phytates from grain by milling. Most of the phytates are present in the outer bran layer, or so they are removed, and this calcium absorption or other functional properties of the grains are improved. So these are some of the example of it. Okay, I think damages to texture during processing. Common example include excessive softening of fruits and vegetables during thermal processing, toughening of fish muscle during frozen storage, firming up bread during storage at refrigerated temperature. When you keep the bread in a refrigerator, it becomes stale, it becomes little tough, hard. Okay, so it should, and this staling increases more below a temperature of 12 degrees centigrade. Of course, it is a reversible phenomena. If you take it out from the bread and heat, then bread may gain its original texture. Emulsion destabilization by heating or freezing, coagulation of sterilized milk during storage, cold shortening of red meats when excessively cooled while in a pre-rigor state, adverse textural changes in tissue foods during air drying, etc. If drying is not properly controlled, even like the fruits, etc., when they are dried, if drying process is not proper, then it becomes very hard after drying. Its rehydration characteristics are adversely affected. So these are, I think, this is whole food technology, which I think some of these we will study in details in various chapters, etc. But I just gave you a summary of these, what are the major desirable and undesirable changes that may take place in the food. So in the quality changes during processing, during preservation, all those things. And it's these, because I feel that you must know, as a food scientist, as a food engineer, one must understand properly what is happening. So where, so it is the same reactions, how these reactions are, what are the factors responsible, which is to increase the rate of a particular reaction or to decrease the rate of a particular reaction. So accordingly, as I told you, in majority of the cases, it is the same reaction which may be desirable in one food, undesirable in other food. Okay. In one condition, in one product, it may not be good. The other product, it may be good. So like that, so you have to use your wisdom, and depending upon the situation, you have to accordingly control the process parameter. I think this was about the quality, etc. I will now devote some time on food safety. But before that, does anyone have any query, any question on those desirable and undesirable changes what we discussed so far in the food during processing, handling, and so on? Does anyone have any query?
So, can you explain what is alkali processing of
Pardon?
soya? Like, sir,
Alkali?
in the improvement of functionality of food, there was something called alkali processing of soybean, sir. What is alkali processing?
Okay, improvement in the functionality of the food, pre-rigor, alteration in the starch gelatinization, alkali processing of soya protein to impart new textural properties. All right, that is here. That is the soya protein after they are extracted. All right, that is basically here. It is the, you can say, the pH control. All right, pH is properly controlled, and it is brought sometimes isoelectric pH is maintained for its functionation, and then there is maybe sodium hydroxide and such other chemicals are used to bring about certain textural its characteristics, realignment, etc., or even some hydrolysis of the protein, etc. Maybe larger molecule may be broken into smaller molecule, etc., so that you get a new soft texture and other things. That is. Okay.
Okay, sir.
Similarly, there are, in fact, yes. Anyone else? Any other question? Okay, if there are no other questions, then I will talk now safety. All right. So far we discussed, I think, I hope you got some good impression about as far as the quality is concerned, quality of a food. Now talk about safety, and let me tell you, safety is the first prerequisite of any food. It is a smaller for the quality. Okay, and safety, they go side by side. A food may be good in quality, but it may be unsafe for consumption. Okay, a food may be safe for consumption, but not good in quality. That can do something. But if it is unsafe for consumption, very good in quality, okay, you cannot afford to take that food. Okay, so that is safety is the first prerequisite of the any food and quality of the food. And
So,
in
can you
broad,
give an sorry to interrupt, sir. Can you give an example of good quality unsafe food?
I am coming. All right, good quality and
Okay.
unsafe food. I will elaborate. Uh, you see that many a times you get a report that the people, so many people died, all right, because of this food poisoning. There was this infection, this intoxication, contamination, etc. There are several examples. Newspapers, and if you search at the internet, these are flooded. So many a time, what happens in that? Maybe in the next class when we discuss with food microbiology, I will little bit elaborate on this. Okay, so these microorganisms, particularly those microorganisms which produce toxin, fine, and
Okay.
these toxin, they are very small in quantity. Okay, or there are certain microorganisms which do not grow on the food, rather they just are present in the food. So food is very good in quality. Its color may be same, its flavor may be same, its texture, it doesn't affected. But that food is is contaminated with the toxin producing bacteria. And when you consume that food, all right, you will like this food. But when you consume the bacteria along with the food or the toxin which is grown, which is produced in the food, it is very in micro level gram, but that is sufficient to cause problem and cause the death of the person who consumes it. So there are many such examples, and in most cases that is canned food, etc. I will tell you in the next. That is, if you read this, whatever written in the blue in the canning industry or another, there are two examples we have taken. Fine.
Yes, sir. Yes.
Okay. Now in fact the thing what you are discussing, that what we say that this food is safe. What does it mean? That the food should be free from any harmful chemical or microbiological contamination or contaminant at the time of its consumption. Mind it. Okay, and therefore it becomes the responsibility of the food manufacturers, food producing company, to issue necessary advisory or guidelines and protect the food in form of a better packaging, appropriate packaging, so that the food, when it is released from the industry, anyway, it is not only that when it is released from the industry, it is safe. No, it must be safe till it is consumed. Maybe 1 month, 2 months, whatever shelf life is there, and that its responsibility becomes, and that is of course governed under the regulation in each and every country. There is labeling requirement, there is other requirement, and but consumers also have some responsibility in this. Like for example, you see that you have got some sort of a fruit juice or vegetable juice, like you say Tetra Pak, etc. Okay, and bigger container you got a 5-liter juice, right? And these juices, etc., they are processed and packaged aseptically, and the processing is done to remove any sort of microbial contamination from that. It is the product which is sterilized, and but the product will remain safe till it is opened. So the industry is responsible for that product till it is opened. But what you do, you brought it into your home, you opened it and just consumed 1 liter, and remaining 4 liter you keep it as such. So for that, you cannot hold the industry responsible. That becomes your responsibility. That is, such products, when they are opened, they should be consumed as quickly as possible, or whatever is remaining, they should be kept safe. Maybe if possible, you reseal or close it properly, keep it in a refrigerator or some other. So that is the concept of the food safety. But let me tell you again, often this concept of food safety is used in its operational sense for practical reasons. And to prove this, I will give you 2 examples. So as I told you, in the canning industry, what is done? Like this canned bottles, you give the canned rasgulla or canned meat or canned vegetables, etc. is available. Okay, so in this, the food is packed in a can. Can is evacuated, it is sealed and then heated. And the very purpose of heating is particularly for the low acid food. It is there to kill the spores of Clostridium botulinum. This thing detail again we will study when we study in thermal processing in this, and I am sure you must be engineering details of this. You must have been taught into the food engineering operations, in thermal process operations, etc. So the Clostridium botulinum is the bacteria which is used as a index organism for the designing of the thermal process conditions of low acid food. Okay, like meat, tea, and such other. products. But this Clostridium, if you see that microbial destruction during heating process, it is a logarithmic decrease. Microbes grow also in the logarithmic fashion, and they are died, they reduce also in during heating, etc. Their killing also is a logarithmic order. Logarithmic decrease means logarithmic means that they you can theoretically never get zero. Okay, so with this, in fact, it is a pre-decided concept that whether you are going to give a 12D process or 5D process or 8D process, etc. in the food, depending upon the type of food, and in some other cases, the type of nature of the microorganisms involved. So 12D process is generally used for low acid food and which are for Clostridium botulinum. 12D means that is 12 decimal reduction. D is the decimal reduction. So 12 decimal reduction process is used for the sterilization of low acid food in the canning. So 12 decimal reduction means that is if there are 10 to the power 12 spores of Clostridium botulinum initially present, then after giving that 12D process, at least one spore will be surviving. So let's take a hypothetical example that there are 10 to the power 12 containers, cans of meat. Okay, and or any other such low acid food. And each can is having one spore of Clostridium botulinum. And this is commercially sterilized, given 12D process. So at least in the end of the process, one can will remain, which will in which the Clostridium botulinum will survive. All right, and the inside the can condition is anaerobic, that is which is favorable for the growth of Clostridium botulinum. Means that is in that one can where the bacteria has survived, it will grow and it will produce toxin. And the toxin produced by this Clostridium botulinum, that is microgram of that toxin, that is 10 to the power -6 gram of that toxin is sufficient enough to kill a healthy person. That is such a potent toxin. So now what is the how to maintain safety? Because this is sealed and it is sold on the market, and but you don't know, nobody knows that which container is having that surviving bacteria and therefore toxin. So there is an advisory that these low acid foods based on the sterilization or destruction of 12D process using Clostridium botulinum must be heated before their consumption, because this toxin which is produced by the Clostridium botulinum, it is sensitive to heat. So if it is exposed to about 80 degrees Celsius temperature for 4-5 minutes, the toxicity of the chemical or toxin, it's nullified. So it becomes safe to consumption. So here it is the concept of food safety with at the type of consumption means that is its operational sense. It has to be taken that food is we cannot say that it is unsafe, but you just heat it and then consume, it will be safe. But there is some other product we take, that peanut butter, let us say a product like peanut butter. How many people, anyone, any idea about peanut butter? How it is made?
Yes, sir. Actually, the oil of the peanut, I guess, is extracted and that is converted into butter, I guess.
Butter. All right, but butter, any idea how much of water it contains?
20%.
About 18 to 20%. Very right. Good. All right, so means that is in the peanut oil, That is, it is mixed with around 18 to 20% water, and then it is made into an emulsion. That is water in oil emulsion, and of course some emulsifier, lecithin, etc. is added, and it is given the shape of the butter. That is also popularly known as margarine or vegetable butter. All right. And this is a healthy component because it contains more unsaturated fatty acid, polyunsaturated fatty acid. So it is considered to be a healthy alternative to the animal butter. But these butters, they are mainly used for as a spread either in the chapati or in the cake, bread, etc., or such other products. They generally used as a spread. So means what I'm trying to tell here is that these butters from peanut oil they are produced, and the peanut, it is a good source, good substrate, which is very likely to be contaminated with a mold or fungi called Aspergillus flavus or Aspergillus niger, and which is very popular or known as mycotoxin producing fungi, that is called aflatoxin. So this is a common problem in the peanut oil, that they may be contaminated, or peanuts may be contaminated with aflatoxin. Okay. So what you can do in the earlier case, we saw the example that the canned food, before consumption, before opening the food or even after opening the container, it is said that you heat it and then consume, it will be safe. But after this peanut butter, before it is used for a spread, as a spread, so it cannot be heated. It has to be used as such. So how to ensure safety in this means that in this case, one has to go back and make sure that the peanuts which were used, groundnuts which were used for the extraction of oil, they are free from aflatoxin, or they are free from aflatoxin producing molds or fungi, Aspergillus flavus. So steps taken to prevent the growth of mold in question may or may not interfere with the retention of some other quality attributes. Nevertheless, the condition of safety must be satisfied, means that it must be ensured that the groundnut peanut which is used for in the processing, that is free from any mold. So here these are, so means that if the food is free from any undesirable or toxic component at the time of consumption, or it is safe to consume, that is, and how we are maintaining this safety, how we are assuring its safety, etc., that has to be taken care accordingly, either following good manufacturing practices, following maintaining good hygienic conditions in the plant, etc., maintaining initial quality of the raw material, or just post processing and packaging, post after the product, before consumption, subjecting you to some sort of treatment, etc. Or so many times the food material we just give it in a microwave before consumption, heat it, so that heating may have the dual purpose. It improves, it makes the food hot. At the same time, it also inactivates if there was any contamination in the food post processing. So that has to be taken into consideration, and safety conditions must be maintained depending upon the type of the food. Okay. What might be the different safety hazards? Like safety hazards refer to those hazards, whether chronic or acute, that may make the food injurious to health of the consumer. So there may be some hazards inherent to foods, like naturally there may be some undesirable components present in the food, like natural toxins, allergens, intolerable ingredients, etc. Okay, natural toxins, like you see the potato. Sometime I don't know if you have seen, many of you, if any one of you are from the village background and have seen while the potato is being harvested from the field, from the soil, so you find some type of potatoes are green in color, and that green, the color of the potato, is because of the presence of certain compounds like solanins, and these solanins are toxic to consume. These potatoes become toxic. Similarly, there are many materials which are having allergens and many biologically active compounds, and the person are allergic to this. So even mushroom is a good example. There are many mushroom varieties, but only 2 or 3 are edible. Other are they have the poison. They are poisonous varieties. They contain poison, but in the appearance they look almost similar. Okay. Then hazards associated with the environmental contamination, like persistent organic pollutants, heavy metals, radionuclides, nucleosides, etc. Okay, like means that is during processing. Sometimes heavy metal, you can see there have been many cases reported earlier in many countries that when the industrial waste water is there in the pond or in the river, and where the fishes, so the fishes when they grow, they are found high in heavy metal content. So these metals come from the industrial waste, and these causes problem. And if you consume this metal, similarly organic pollutants or even radionuclides, they may contaminate the food, etc. So the environmental pollution becomes a problem. Then similarly, hazards arising out of the microbiological activity. There are a large number of these bacteria, viruses, bacterial toxin, biogenic amines, mycotoxins. These are the various compounds which arise post-production, or even sometimes pre-production, also pre-harvest or post-harvest. That is when food comes in contact with certain microorganisms, the microorganisms grow and produce certain toxins, etc. Similarly, there may be hazards emerging from food processing itself, like acrylamide, chloropropanol, etc. Acrylamide, you see, it is one of the major problems in the ready-to-eat snack food industry. Even in the bread, the crust of outer dark color crust of the bread, it is high in acrylamide content. Fried potatoes, French fries, etc. They are very high in acrylamide. So this acrylamide, when such foods are heated, maybe generally above 120 degrees Celsius temperature in the deep frying or in the baking oven, etc. Then asparagine is an amino acid, and sugar, reducing sugar, they combine, and this is a Maillard reaction, non-enzymatic browning reactions take place, and acrylamide is formed. So this acrylamide formation is a major challenge in such industries, and that is one reason why these foods many times fried foods, etc., which are called sometimes as this junk food, etc. So one has to, because lowering the temperature may not be always, it may create other safety problems, etc. Similarly, chloroprophenols also, these are oils, etc. In oil milling industry, due to heat, etc. These undesirable compounds are formed. So these are some of the safety concerns following the food processing, like in the case of safety of bottled water, because water is a good source for contamination, microorganisms easily. So when you are taking bottled water, so one has to see that what is the water source, piping, treatment process and bottling equipment, packaging, even quality control systems should be proper analysis, etc. Similarly, safety of the soft drinks, if you see, microbiological contamination, packaging material, chemicals, other additives, equipment used in processing, formation of mutagens, carcinogens like nitrosamines, or even the many of the processed food. Trans fat is a very, very even important consideration, particularly when the food is containing fat, and the fat in the unsaturated fatty acids, normally these fatty acids unsaturation bonds, there are the cis position H. which are present. And when these oils are heated, the fats are heated, during heating, cis fat or cis oil is converted into trans fat. Okay, and intake of trans fatty acids from partially hydrogenated vegetable oils or such other products, even in the that is the refined oil, hydrogenated oil, in the fried products, etc. They contain more trans fat, and this is a very, in fact, trans fat are much harmful than those of the saturated fat, because these trans fatty acids, they are not excreted from the body, rather they are also not absorbed. They just go on depositing inside the your arteries or veins, etc. So inside there is a, you will find after some time, if you consume regularly with the trans fat containing products. So this interior, they get an interior of the artery or veins, they get deposited, and therefore passage for the blood flow either to the heart or from the heart, that is both may be blocked, and it will become, and it result in the cardiovascular diseases, etc. are okay. So they are much of a problem. And even in this trans fat, many countries, there is a very strict regulation on trans fat. Countries like, there is company like McDonald's, etc. They were sued in Australia or many other such companies because they were making the claim that their product is trans-free, but it is not. It is really not possible. If the fat or oil, if it is heated, no one can control, no one can say that it is free from trans. So in many countries, there is some labeling of trans fat. Maximum trans fat permitted in few countries is 2%. Even in some countries, it is 1%. And so that is, so these are the some of the concerns. Right. So here a combination, you see quality and safety. This graph shows that process severity, how. So if you give a, by having a proper processing condition, more or less severe a process treatment, you can have a better, more or less quality or more or less safety. Generally, it is seen that if you like heating, is more severe heating, if you use, its safety will be good, but the quality will go down because this heat will adversely affect the quality nutrients as well. So there are many other processes as well. So one has to go for similarly non-thermal process. If you say non-thermal, alternate thermal process, etc., that they are good in quality, but their safety is still a big question whether they are safe to consume or not. Only time will tell, because the thermal, at least thermally processed food, they are time tested, and they can be relied as a safe food. But as a food processor, one has to make a balance, and accordingly we optimize the condition, etc., and find out is up to what level the process should be used, so that the food is good in quality. Otherwise, food may be safe. If it is not good in quality, if its nutritional value is completely lost, so what is the purpose of eating that food? Similarly, the food is very good in quality if it is not safe, as I told you earlier. So that a proper balance has to be made, conditions has to be optimized properly and used suitably. So this is all. I think I will take up a 2-3 minutes break here. That break we will use for making, if anyone is having any questions, etc., let us have. Okay, we can devote 2-3 minutes time to take a few questions of so far whatever we have discussed. Yes, anyone?
Sir, can you please explain the peanut and aflatoxin, that thing in detail again? I was unable to understand the relation between that. You told something about molds and then aflatoxin. I could not understand the relations.
थोड़ा अपना नाम बताओ कौन बोला? अभिलाष मुखर्जी.
Yes, sir.
Okay. That you see, that aflatoxin is again a type of compound, chemical compound, you can say biochemical compound, all right, and which is produced by the fungi mold. It is a bacteria. Sorry, not a, it is a some sort of microorganism mold. In the next class, we will take up what are the different types of microorganism and which how they grow, etc. All right. So this fungi, if the peanut is contaminated with the fungi, a mold, which is Aspergillus flavus or Aspergillus niger, then this fungi will grow in the peanut and it will produce the toxin, which is called aflatoxin. Is it clear now?
Yes, sir. Yes, sir. So this microorganism produces this aflatoxin.
Yes, microorganism, that fungi, fungus.
Hmm.
Okay, that is fungi. You see, you might have seen that bread or even any other food, even some material, if you keep in a damp weather, bread, then where there is a white cottony growth of mycelium, cotton-like growth you find in the bread, we say that it is a fungi. So this is
Yes.
a type of microorganism. So it is a similarly, that is a type of fungi, is Aspergillus flavus or Aspergillus niger, and which produces aflatoxin. And this aflatoxin also, maybe immediately it is not that poisonous as like the toxin produced by Clostridium botulinum, but in the long run, this aflatoxin also create problem. So the product of peanut butter has to be how the safety of peanut butter is maintained. That peanut which is used, it is free from aflatoxin or aflatoxin producing bacteria. Clear?
Yes, sir.
Okay. Anyone else? Any other question? Good. Other people also, please. Shruti Maheshwari, or what name? Shruti Mahalakshmi. Ariel, are you here in the class, Shruti?
Yes, sir. I just have some issues with the mic.
You have some issues earlier also. Carl, you tell what did you understand so far? Anything can you tell? Give me a example where the processing improves the flavor of a food. Shruti? You are again muted. Hello. Hello, it is a good excuse. The network is not a problem. Network is a problem. Okay. Haji, Raghav Sharma. Raghav Sharma, can you open, unmute yourself and speak? You are muted. Good. क्या हो रहा है? Only I am speaking and none of you are responding. Are you not there in the class or what? What is happening? These names I am calling. Yes. Bhanvi Suryavanshi.
Hello.
Yes.
Yes, sir.
Yes.
Yes, sir, I'm present.
Can you tell that what we give me an example where processing improves the texture of a food?
Mm, Sir, maybe coffee beans.
Tell me.
Hello.
I'm talking. Yes. Please tell. I'm talking the texture.
Hello.
Yes.
Boiling of rice.
So I think all of you please make it a point and try to respond to the queries, because if you are and try to understand the thing, there is no point I go on explaining and reading the PPT, explaining the things, and if you do not follow it, if you do not understand it, no, purpose is defeated, purpose is not served. So at this we cannot, until unless we get a response from you, because you people are not opening your videos, I cannot see you, and if I ask, you are not responding. Be serious to yourself. If you are really interested in studying the subject, gaining some knowledge, it's my humble request to you all. Please be serious and respond to my queries. Respond when I ask. At least when you speak, it gives, at least it becomes encouraging. I also, otherwise it becomes anyway boring and monotonous, and I am going on speaking, no response. So that you have to make it lively. And we can make it lively by more interaction among each other, by interacting with us. What is happening? Are you not finding it interesting, the subject? Can anyone tell?
Sir, till now, like it was more of biology stuffs. And basically,
Yes, sir.
personally, I hate biology because there is more of remembering here.
Okay. Anyone else? Because the food is a mixture of chemistry, microbiology, or engineering, all these things. And here, obviously, in this subject, you will have more that science. Rather, it is a science-based course. Engineering will be taught in other subjects, but in the latter part, when we talk about processing, etc., right? There also some of the science, etc., will be taught. So it is not you cannot say the biology, but yes, science of the food. But mind it, you are agricultural engineer and biological engineer, and if you see that I hate biology and science, I think that is not a good thing. You have to start loving it. You have no option if you want to be in this profession. ठीक है, Jaychandra?
Yes, sir, I'm putting my effort to it.
You see, there is a saying that you should always try to marry a girl to whom you love. But if you are not able to do that, start loving the girl to whom you have married. That is a very old saying. So since this subject, okay, agar biology mein tumhe that, you should have gone to mechanical engineering, you should have gone to computer science and all those things. But you came to this department, and that is why it was your whatever your option choice. So once you have come to this department, I think you have to start loving the subject and tell us what are the things, how you can generate interest, and what can be done. You put your own effort, you tell us, we can also help you. All right.
Sir, if you could share the slides just one day before, we can refer to it and come to that.
Slides, it cannot be shared one day before. I have already told you the books. You know that what are you read the books. Okay, don't try. I cannot make it a spoon feeding. Read the books, or even these PPTs, etc. If you in the team in the earlier classes also, these lectures are already recorded. So we cannot do that type of spoon feeding that will give you one day before the slides, and at least you come before, read the books. All these things are available in the book, and I have already given the book to you, uploaded the book in the class study material.
Yes.
Yes, do some effort. Not like this that everything will be given in the prepared form to you. We are just facilitator. Learn tumhe karna hai, and if you want to learn, you have to put effort for that. Yes, anyone else? Good, at least I'm happy. Good to know from you that this take a tell your frank opinion so that we can make it more interactive, more lively.
And also, sir, can you please when you are talking about the different things, na, sir, you are mentioning all of them. So, sir, if you can take some time and at least explain one, then, sir, we can ask some questions and all. And but if you keep going through all the processes,
That's right. That's what. इसलिए तो थोड़ा सा what you do. I don't mind that you just raise your hand and tell me. Yes, you unmute yourself and ask the question. Wherever point is there, whenever I am talking, you ask the question. We will interact. That's true. That's good. Because, all right.
Sir, actually, means, sir, when you go faster, it's like very abrupt to interrupt. So that is why, sir. So, sir,
Then
like if you
that's
can
why
like.
इसमें तो I am giving after 15-20 minutes discussion. I'm giving you just 2-3 minutes to ask the question. But when I am telling at least only you are asking 1 or 2 questions, but nobody else is responding to that. Nobody else, even I am calling the name, people are not responding. It means that simple say that they are not here, just they have joined the class and they are not attending the classes. Right? Yes, I fully agree. Wherever I don't mind you interrupt in between. Let us discuss the things. Okay,
Okay.
but I think I would like that is other people also should respond. Fine.
Okay, sir.
So can I move further now, or there is any other? Jayachandra, tell
No,
me.
sir. No, sir. Nothing. You can.
Can I go ahead?
Yes.
I hope now you will start loving biology.
Sir, it's not like that. I'm trying to grasp as much as possible.
You try to grab some big part, and we are there to help you. All right. But in fact, the statement which you made that I hate biology, कोई भी चीज hate करने का नहीं है. जो तुम्हारे ऊपर एक compulsion में आया है. So even what you might be doing in other subjects, etc. यहाँ तो फिर भी it is a blend of science and engineering both. So if you are here in what about the other subjects which are purely like crop science and other things? What you will do there? Okay, try to understand. Okay, okay. So now I think can you see here?
Yes, sir.
So far I gave you understanding food, its quality, its safety, different reactions. All right. Now I will give you what are the different variables, different factors which influence these reactions. Okay, and therefore these factors become the important point of control during processing or preservation and all this. Okay, and the major factors are temperature, time, and the rate at which dt by dt means rate at which temperature changes with time. Then pH, composition of the product, initial composition of the product at the time when it is being processed. Then composition of the gaseous phase, means environment in which the food is being processed, or environment in which the food is being kept post process, during storage, during handling. And last but not the least, more important, you can say most important, is the water activity. That is water which is the present in the food, and its form, in which form the water is present in the food. So these are some of the major factors. All right. So I will elaborate one by one each. Okay. So temperature, Jay Chandra.
Yes, sir.
Yes. Now, can you tell what is that K is equal to A e to the power minus EA by RT?
Sir,
What is this reaction?
sir, it is rate, like
What is that rate?
rate
Explain. I think you, it is written also on the PPT.
Arrhenius equation.
Arrhenius equation. What does Arrhenius equation say?
It says that rate constant is dependent on these factors like temperature and uh,
What is EA? What is EA? Now you will say that, sir, I hate mathematics also.
no, sir, no, sir.
Then what is R and T? Yes, tell me, it is not a very big thing. It is very preliminary. Even in the schools also now this is told. Okay. That is this is the reaction which in stage the how a rate of the reaction or a rate of a particular process will be influenced with the change in the temperature. Okay.
Sir, EA
In a
is
pardon?
activation energy and
EA is activation energy, R is the gas constant,
yes,
universal gas constant, T is the absolute temperature in degree Kelvin, etc. All right. And
yes,
this is a very, very well established relationship that is given by the Arrhenius equation, and which gives a chemical reaction. That is the effect of temperature on a chemical reaction rate. Fine.
yes,
In
sir.
the food also, there are various chemicals. There are various biochemicals, etc. Biochemicals are also, there are chemical only. So I will come in the next slide. But what I am say that is, you told the data conforming to the Arrhenius equation yield a straight line when log value of K is plotted versus 1 by temperature in degree Kelvin. All right. This is the Arrhenius relationship, well established. But what is important here now, I am trying to tell you, in the food also, it is a biological material. In the chemicals are present biochemicals. So in this, when you process a food, there are changes in the constituents and chemical and environment, etc. of the food, and therefore this Arrhenius relationship, this conforms to the food, in the food, only over a certain intermediate temperature range, not abruptly. In the chemical reaction, you can say yes, it is that is a relationship food, but Arrhenius relationship in the food holds true only over a certain temperature range. So if you are modeling a food, if you are modeling a process, if you want to apply Arrhenius relationship to develop a model temperature, how, then you can not blindly, but rather you find out first what is that temperature range. All right.
Yes, sir.
Okay. So here you see that figure. In the figure, that is it is explained. There are a non-enzymatic reaction and a enzymatic reaction. Okay, the top one is the enzymatic reaction and bottom one is a sorry, top one is the non-enzymatic reaction and bottom one figure is a enzyme catalyzed reaction. So we have taken the two examples to explain you that how and why this arrhenius relationship doesn't hold true in the case of food universally. So when the food is being given, that is you see in the figure, there is a point on the top of this figure in non-enzymatic reaction, and the bottom there is a B. Okay, and from this A to B, you can say only this is the range. This it is following the conforming to the arrhenius relationship. So above that certain temperature, okay, on lower temperature side as well as on higher temperature side, there is deviation from the linearity. Okay, so A says that above a certain value of T, there may be deviation from linearity due to change in the path of the reaction. That is the maybe the medium may become more concentrated. Okay, substrate which is there in the reaction, it become its concentration may change, more concentrated, less concentrated, solutes availability, etc. So the path of the reaction may change. Similarly, the B point you see, that is the when the temperature is lowered below the freezing point of system. Let us say in the freezing process. All right, what happening in the freezing? When it's lower below the freezing point of the system, then the ice formation, that is water, converts into ice. So as the freezing process progresses, the amount of water goes on diminishing, decreasing, but the solutes present in the water, its content goes on increasing, because earlier, let us say earlier, they were in large amount of water, the same solutes. But when the water is freezing, a point will come that the solute concentration of the solute will become highly dense. Water becomes less, solute becomes more. Okay, and this change in the concentration of solute in the unfrozen phase can either decrease, that is supplement the effect of decreasing temperature, or increase the reaction rate depending upon what are the different types of different solutes, etc. which are present. Okay, so there may be deviation from the linearity. Okay. Similarly, if you take the at the bottom part of that reaction, there is it is a enzymatic reaction, enzyme catalyzed reaction. Then the same thing, C and D, it is shown. The point C, if you see that it is an enzymatic reaction above a certain temperature. When you heat, there is a denaturation of enzyme. There is the enzyme structure is changed, enzyme activity is lost, and therefore the enzyme may become completely inactive. Similarly, in the lower temperature side, in the vicinity of the freezing point of the water, subtle changes such as dissociation of the enzyme complex, rate of dissociation of the enzyme complex can change. There may be shift of the active site of the enzyme from one place to other place, and because of this, again the reaction rate declines. So the major takeaway from this is that Arrhenius relationship, Arrhenius equation cannot be blindly applied in food system for a long range, but one has to experimentally, there are procedures by which you can find out that relationship, that is what is that temperature range over which the Arrhenius relationship holds true. That RNS relationship of food system can be used only over. I already told this thing. Okay, and to summarize, the deviation from the RNS relationship can occur because the enzyme activity may be lost, the reaction pathway may change, or may be influenced by a competing reaction. Okay, the physical state of the system may change, or one or more of the reactants may become depleted. So there may be a number of these changes, etc. Okay, and because of that, RNA. This point. Okay, at this point, Abhilash Mukherjee.
Yes, sir. Till now it's okay.
Okay, fine. Thank you. All right. Now, next is the time. Okay, and time is the temperature you saw that again time is an important factor to be considered together, and this rather should be considered with the rate at which temperature with the changes with the time. Rate at which the temperature changes with time, because what happens during storage of a food product? What do you want to know? That yes, we want to know the capability of food with respect to its quality. That is, we want to know that what the focus is on the time with respect to integral change of chemical and microbiological changes that occur during a specified storage period. Okay, and these chemical and microbiological changes, obviously, they dependent on both on time and temperature. And if you change the time, change the temperature, the rate will be high, rate will be low. So in a small period, the food will spoil. In a small, it may become a long period. It may take to get the spoil. So ultimately, we want to know that yes, the what is the time during which the food is above a certain level of quality. And that is the integral of time and temperature. So the rate during the processing attention is given primarily to the rate because it's a variable, time variable, as it appears in the rate expression. And this relationship is important because it determines the rate at which microbial reactions or biochemical reactions are influenced, how the microorganisms are destroyed at the relative rates of competing chemical reactions. Okay. Time can also have an important influence on the relative concurrent reactions. For example, if a food can deteriorate by both a lipid oxidation and a non-enzymatic browning, okay, and in both the reactions, the lipid oxidation reaction, that is the triglycerides are oxidized into various carbonyl compounds. Similarly, in the non-enzymatic browning reaction also, I told you sugar and amine, they interact with each other and form a new compounds. And the end product of this, many times it may so happen that the end product carbonyl compounds which are formed as a result of non-enzymatic browning reaction process, they may be having antioxidant properties. So if these two reactions, lipid oxidation, enzymatic browning, both are taking in a food, okay, and if the browning reactions products are antioxidant, it is interesting to know that if there is a overlapping, sufficient overlapping in the both processes, so the browning reaction product may influence the rate of lipid oxidation. So that is how the rate is taken. Then third factor is the pH. pH influences the rate of many chemical and enzymatic reactions. Extreme pH values are usually required for severe inhibition of microbial growth or enzymatic processes. Okay. Even all these reactions, whether microbiological reactions, biochemical reactions, enzymatic reactions, they require certain optimum pH for their growth. And beyond that, whether you go towards the lower side or you go towards the higher side, the reaction change, and the reaction will be either generally that the pH influence is the bell shape and optimum pH, and then reaction stops in both the sides. So these conditions can result in acceleration of acid or base catalyzed reaction. In contrast, relatively small pH changes can cause profound changes in the quality of some foods like mussels, etc. Okay, so that is even rigor mortis, etc. That is, it is the post slaughter in the case of animal flesh. That is how the glycosis takes place and how the pH is developed, and it influences the textural characteristics. And sometimes if pH becomes very low, then muscle become very hard, and that is called rigor mortis. So these are various complex reactions take place. I will not go into. So these were the pH. Then finally composition of the product, because the initial raw material quality, this determines the reactants which are available, that are available for chemical reactions. The relationship between the composition of the raw material and composition of the finished product is very, very important from quality point of view. If the raw material itself is of not a good quality, you cannot expect good quality of the end product. By processing, you cannot do much. For example, there are, suppose, the handling of fruits and vegetables post-harvest. Okay. That if the temperature and other conditions during storage, if they are not properly maintained, it can affect the degree of browning during dehydration and deep fried frying. Let us take an example, potato. Potato, it has starch. Starch is, it is amylose and amylopectin, which is the glucose is the resulting monosaccharide. So now this potato, if it is during storage, normally it is recommended, I think, 3 to 4 degrees Celsius temperature in the refrigerated. Refrigerated cold storage, it is maintained to store the potato. But very important is that potato, whatever temperature is maintained, it should be constant. If it is 4 degree, it should be, effort should be made to have 4 degree constant temperature in the storage, cold storage. If there is a fluctuation in the temperature, then these fluctuating temperature conditions causes hydrolysis of the starch. Okay, and starch is converted into glucose, and under this low temperature condition, even glucose is converted into, by enzymatic reactions, it is converted into fructose. So that cold stored potato, that is, it was stored in a environment where with the fluctuating temperature, it will have more amount of high content of sucrose, glucose and fructose, etc. And therefore these glucose and fructose, they are reducing sugars during heating process, during drying process. They will interact with the amino acids together and cause browning. So the manner in which the post-harvest the fruits and vegetables are handled, it can influence that the presence or absence of, that the polysaccharides may be converted into monosaccharides, and these monosaccharides may cause problem, a darkening of the product during deep fried frying or drying, and etc. Potato chips prepared from such potato will become darker in color. If during storage or handling there is sufficient hydrolysis of starch, the potato contains more amount of glucose. This glucose is bound to cause browning. Similarly, handling of animal meat post-mortem influences the extent and rate of proteolysis, glycolysis, etc., which affects its texture, flavor, color, and so on. Okay. That's why, when in fact after the animal is slaughtered, a control rate of glycolysis should be properly cooled down so that the glycolysis at least continues for some time. Okay, and there is a proper development of color and flavor. So muscle, it gets a desired texture, flavor and color too. So that is very important. So there are many other cases, like even blending of raw materials may cause unexpected interactions, right? Rate of oxidation can be accelerated or inhibited depending upon the amount of salt present in some of the food. Salt are added, in some cases sugars are added, or in fabricated foods, the composition is controlled by adding approved chemicals like acidulants, chelating agents, antioxidants, etc., or certain undesirable components are removed from the mixture. Okay. I gave you the example of removal of glucose from dehydrated albumin and also. So these also means there is the initial raw material quality, what are the various components present, amount, it will influence the processing parameters, processing conditions. Then composition of the gaseous phase. That is, gaseous phase means influence of oxygen as a reactant. Okay, in situation in which it is desirable to limit oxygen, it is almost impossible to achieve complete exclusion. To detrimental effect of even a small amount of residual oxygen becomes very evident during storage and in the product like coffee, etc. You see that coffee, when it is packaged in the vacuum, it maintains the quality for longer time. But if you go by vacuum processing by mechanical means, etc., so at least a small amount of 1% or a small amount of oxygen remains there in the packet or head space gases in the package, and that small amount of oxygen is rather more detrimental, and it results in the stalling of the coffee. Coffee loses its flavor, etc., in very small time. Okay. Also, so what is the practice that yes, care should be taken that now the most of the vacuum processing methods are being replaced by nitrogen flushing, because nitrogen flushing keep the inert environment and oxygen effect of a small amount of oxygen is nullified. Early formation of a small amount of dehydroascorbic acid can lead to browning during storage, etc. Okay, now the rate of reaction, microbial reactions, etc. The presence of oxygen, absence of oxygen, or even carbon dioxide, oxygen content is decreased, and carbon dioxide content in the atmosphere is increased, and that is the basis of the control and modified atmosphere storage. Okay, in the relative humidity, other things are. Okay. Then finally, the water activity. Water activity is the one of the most important variable that control reaction rate in foods. And important factors in enzymatic reactions, lipid oxidation, non-enzymatic browning, sucrose hydrolysis, anthocyanin degradation, etc. Can anyone tell what is water activity?
Sir, I can't define, but I can explain.
Yeah, just please explain. Please explain.
Like it is the free water molecules which are available for reactions.
Free water molecule, I think yes, it's good concept. But you see, A is equal to AW is equal to PVI P0. That is it is the water pressure, vapor pressure of the food at a particular temperature divided by this saturated vapor pressure. Or in other words, you can say the equilibrium relative humidity ERH. All right, expressed in decimal. That is, suppose 85% is the equilibrium relative humidity, so 0.85 will be the water activity. That's what. RA, which you told rightly, that is, it is the free water that is available, that helps, that determines the water activity. That is the free water present in the food, which is available for chemical reactions, microbiological reactions, and that free water gives its water activity. But when we want to express water activity, in certain value, that is pure free water, which we find in the sea, in the pond, lake, etc. It has water activity of 1. And when any solute is dissolved, solutes, then it depresses the water activity. More the solute concentration, less will be the water activity. So most of the food, and this water activity means that is the lower the water activity, means that is the less free water, and this water which is available, it is less available for the microbial reactions. Now there are, I don't know whether we will get time in this subject to read that. Otherwise, there are high moisture food products where the moisture is kept as high as 95, 96%, 98% in some cases, but still they are made safe because this moisture is bound in certain forms. HMFP product. You see that most of the intermediate moisture food products, they have water activity in between 0.75 to 0.85. Okay, that is the jam, jelly, or such other products which you have, means that is they are below 0.75. Almost most of the undesirable microorganisms, spoilage causing microorganisms, their growth is suppressed. They cannot grow. Only a few yeast and molds are high salt tolerant fungi, they can grow, and that too up to 0.6. So if you can say lower down the water activity to a level of 0.6, all right, the food will become particularly safe and it will not support any chemical, microbiological, and other reactions. It will become room temperature stable. And that is all the aim of the modern food technology is, that you lower down the water content of the food, reduce its water activity by one or the other method. What are the ways of reducing the water activity? Either you remove the water by evaporation, concentration, etc., or add some salt, sugars, etc., to bind the water. Water will be present in the food, but it will be in the bound form. Or in some cases, both are done, partial removal and partial binding, and water activity is controlled. Is it clear?
Yes, sir.
Yes.
I have a question. Like,
Yes, yes,
how
please.
is freshness determined? Is it on the basis of moisture content present or
Pardon?
freshness of a material?
Freshness of the food. Maybe moisture content may be one parameter. There are some other parameters also, particularly if you talk about freshness of the fruits and vegetables. All right. Then it's a turgor pressure inside. That is the moisture which is present inside the cell material. How much pressure is exerting on it, and then the texture of the. Then there are gloom, color, flavor. So there are various parameters. All right. But moisture content, you can say, is it may be one important parameter on the basis of which the freshness can be, because if the it deteriorates and it becomes wrong, the moisture content, depending on the condition under which it has been handled, it is stored, moisture content may be more or less depending upon the type of food. That is, if it is a processed foods, dehydrated foods, okay, it can regain moisture. It is a hygroscopic material. Or the fresh fruits and vegetables, it may lose moisture. Clear?
Yes, sir.
So sir, suppose fruits and vegetables, they would have the most amount of water activity at the beginning, right? I mean,
At the beginning, yes.
so sir, then we have seen here that what high water activity means more susceptible to, I mean, microbial interventions. So
Yes.
sir, that would mean that the fresh food would be the least safe to eat and processed food would be the most.
Yes, that's you can say so. All right. But that's why fresh fruits or vegetables, etc. All right, when they are taken They say that you should clean it, wash it, sanitize it, because most of the fruits or vegetables, they are provided with the protection, that is the outer peel. All right. And the husk, etc., and the grain, they are made in such a manner that if it is intact, not many microorganisms can penetrate into it.
Okay.
Right. So, but if there